Welcome to today's lesson on enzymes and digestion. We will be covering the activity of some important enzymes involved in the digestive system. These are amylase, proteases, and lipases. We will also look at how the products of digestion are utilized in the body. In our lesson today, we will focus on learning where important digestive enzymes are produced in the body. We will also look at how they act in the digestive system. We will then explore how the products of digestion, such as glucose, amino acids, and fatty acids, are used by our body to maintain health and support various bodily functions. So far, you have covered the basics of the digestive system, including its structure, the general process of digestion, and how enzymes operate in order to speed up the rate of metabolic reactions. To begin our lesson on digestive enzymes, let's recall why we need them. The food we eat is made up of large, insoluble molecules like starch, proteins, and fats. These molecules cannot pass directly through the walls of our digestive system into our bloodstream, so they need to be broken down into smaller, soluble molecules. This is where digestive enzymes come in. They catalyze or speed up the breakdown of these chemical bonds, making the nutrients available for absorption and use by our bodies. There are three main types of digestive enzymes you need to learn about: carbohydrases, proteases, and lipases. Each has a specific role in breaking down different types of molecule. Carbohydrases are enzymes that break down carbohydrates into simple sugars. One important carbohydrate is amylase. Amylase breaks down starch, a complex carbohydrate, into maltose, a simpler sugar. It is produced in the salivary glands, the pancreas, and the small intestine. Maltase, another enzyme, then breaks down maltose into glucose, which is readily absorbed into the bloodstream. Proteases are enzymes that break down proteins into amino acids. Different proteases act at different sites in the digestive system. Proteases break proteins into smaller peptides, and ultimately into amino acids. They are produced in the stomach, the pancreas, and the small intestine. Pepsin is a type of protease that breaks proteins into smaller polypeptides. Other proteases that are produced in the pancreas and small intestine complete the breakdown of polypeptides into individual amino acids. Lipases are enzymes that break down lipids, such as fats and oils, into glycerol and fatty acids. They are produced in the pancreas and the small intestine. Before lipases can effectively break down lipids, bile emulsifies the fats into smaller droplets. This increases the surface area for the lipases to work on, speeding up the digestion process. After digestion, the products, such as simple sugars, amino acids, glycerol, and fatty acids, are absorbed into the bloodstream and used in various ways. For example, some glucose that is released from the breakdown of carbohydrates is used in respiration to release energy. From protein digestion, amino acids are used to synthesize the new proteins that are needed for growth and repair in the body. Glycerol and fatty acids from the digestion of lipids can be used to make new lipids for cell membranes, or they can be stored for energy. In addition to breaking down molecules, enzymes play a crucial role in building them up. For example, enzymes are involved in synthesizing larger carbohydrates from simpler sugars. One case of this is when glycogen synthase, a carbohydrase enzyme, helps to form glycogen from glucose. This serves as an energy storage molecule in animals. Enzymes are also important in catalyzing the joining of amino acids together to make proteins. Finally, multiple enzymes are involved in synthesizing lipids from fatty acids and glycerol. Before we conclude this lesson, let's consider the history of digestion, a process that has intrigued humans for centuries. Ancient Greek physicians, such as Hippocrates, believed in the concept of humours, where they thought digestion was like cooking food in the stomach. They imagined that different humours, or fluids in the body like blood and bile, played a role in breaking down food. This early idea was a far cry from our modern understanding, but it shows just how long we've been curious about what happens after we eat. To summarize, digestive enzymes are essential for breaking down large, insoluble molecules into smaller, soluble ones that can be absorbed and utilized by the body. Amylase, proteases, and lipases each have specific roles in this process, and their proper functioning is crucial for digestion and health. The products of digestion are valuable resources that our bodies use to build new molecules and release energy, highlighting the importance of this intricate biological process.
Watch the rest of the GCSE biology course at Sophos Education.